compare the movement of molecules across the membrane by the carriers and channels. So we're really speaking about facilitated diffusion. Facilitated diffusion. And facilitated diffusion is somewhat similar to simple diffusion in that molecules are moving down their concentration gradient, but as the name implies, facilitated, this requires some sort of help, some sort of mechanism. Okay, these molecules are either not the right shape or the right size, or they are too large, or they're polar, and so they're not going to go through the membrane on their own. They're going to require the help of a transmembrane protein. That transmembrane protein we call a carrier or a transporter protein. Okay, and some of the characteristics are the protein is transmembrane, which means it spans the entire lipid bilayer. If you remember those general characteristics we talked about with membrane proteins, we said that some were one-sided and some were transmembrane. This is an example of a transmembrane protein. It's going to go through the entire width of the lipid bilayer. Another characteristic is that it has a specific binding site for certain particles or certain molecules. Okay, so it's only going to bind glucose, for example, or it's only going to bind sodium, for example. So the binding is very specific at the binding site. Another characteristic is that binding only occurs at one side. And when we talk about different proteins, we'll see some that have binding taking place on both sides. For a carrier protein, binding only happens at one side at a time. All right, let's look at this example using the transport of glucose. So we have the cell membrane, or the plasma membrane here, okay, which is comprised of this lipid bilayer. We have the extracellular fluid and the intracellular fluid. And embedded in the membrane of this cell, we have this transporter protein, this glucose carrier protein. Now, we said already, glucose is not optimized to go through the membrane using simple diffusion. So it's going to require this transporter protein to bring it across. Now, it's going to bind on the binding site that is exposed to the extracellular environment or the extracellular fluid, where glucose is usually present. And then that protein is going to go through a conformational change or configurational change. It's going to basically shape shift. And when it does that, it's now going to expose the binding site to the intracellular surface or the intracellular environment. And that is going to facilitate glucose going through that membrane. Okay? So important things to know here is that it's facilitated. It's still going to go according to the concentration gradient. Okay, but it's going to require some help, and the help is a carrier protein. Let's talk about factors that affect the rate of facilitated diffusion. Firstly is the rate of transport of each carrier. Each carrier, different carrier slot body, different cell types, different functions, have different rates of transport. If we take the GLUT4 transporter, for example, it can transport 1,000 or 10,000 rather, glucose molecules per second. 10,000 glucose molecules per second come across the membrane from the GLUT4 transporter. So the rate of each transporter will determine the rate of facilitated diffusion. And that differs by tissue types, by cell types. Secondly is the number of carriers in the membrane. Okay, the number of carriers in the membrane. Now, a cell can upregulate or downregulate the number of these carrier proteins that are embedded in the membrane. For example, let's take that same GLUT4 transporter. If you just had a large meal and you're actively trying to absorb glucose into your cells, the cell will embed or upregulate to embed more GLUT4 transporters into the membrane so you can bring across more glucose. If you're not so full, if you're not trying to bring glucose in, those GLUT4 transporters will downregulate and they will not be embedded in the membrane. And so this can change the rate of facilitated diffusion for glucose. The third factor is the concentration gradient. The concentration gradient. And just like we saw with simple diffusion, if we have more of a gradient present, we're going to have more transportive molecules. But the difference with facilitated diffusion is that we do not have a continuous linear graph into infinity. So let's look at the difference with this graph and simple diffusion. All right. So for simple diffusion, we said that the net flux would increase as the concentration gradient increased. And that would go on into infinity. If you had more of a concentration gradient, you had more movement of particles. For facilitated diffusion, if you had more of a concentration gradient, the net flux or the net movement of particles would increase to a point where that graph begins to level off. Okay, why does that graph now level off? Why do we have that graph leveling off, or that curve leveling off? Why doesn't it increase proportionally the way simple diffusion did? Can anyone think of that? Good. Good. Perfect. So you have exhausted or used up all of your group 4 transporters or whatever transporter, and so there is a leveling off of the net flux or the movement of particles. And this basically means that regardless of how much the concentration gradient increases, we have a limiting resource here. The limiting resource is that transporter. You cannot bring more molecules across the membrane than you have active free transporters to do so. Okay. Okay, so let's look specifically at some of the details of this example, the group 4 transporter. Now, it is a molecule, it's a protein that is synthesized in the rough endoplasmic reticulum, which simply says to us, right, there should be a review, that if it's synthesized in the rough ER, it's made for the membrane of the cell or to be exported. Okay? If it were made on a free ribosome, it would be used for cellular purposes to remain inside the cell. So then we pack it into a vesicle. It's going to be transported to the membrane and embedded into the membrane of the cell. Okay. Now the presence or absence of insulin is going to stimulate or inhibit GLUT4 insertion into the membrane. So in states where the insulin is increased, let's say you had a meal, you spiked your insulin, lots of insulin will stimulate more GLUT4 transporters to be embedded or inserted into the cell membrane. And that would facilitate more glucose uptake. Now, if you think about states like, or diseases like diabetes, where there's a lack of insulin, there is less GLUT4 transporters or less GLUT4 carriers inserted into the membrane, and so we cannot uptake glucose as readily as normal. Alrighty, now in both types of diabetes type 1, where your body does not make insulin, you have a hard time bringing glucose in, as well as diabetes type 2, where you're insulin resistant. Although your body's making insulin, your cells are not receptive to that insulin. And so in both scenarios, you don't have the GLUT4 protein, you cannot bring in glucose, and you get the signs and symptoms of if there is no glucose present, although there is glucose in the body. Okay, so that was facilitated diffusion. Let's move on to talk about diffusion through channels. Okay, diffusion through channels. Now, this is also a form of passive transport, but it's going to happen through an opening or a pore or a channel that is embedded in the membrane. Okay. So these channels differ in some regard from the carrier proteins that we spoke about earlier. They are also transmembrane proteins. They function like a passageway or an opening. And so if you look at this image here, we can see the lipid bilayer. And then we see this opening that's like a little passageway or a little tunnel that allows the passage of certain molecules, usually charged particles or ions. And they are substance specific, meaning that only a specific substance can bind or can traverse this pore and end up on the other side. Now, there are different types of channels. There are aquaporin channels, which specifically transport water across the membrane. And these are usually seen in the large intestine as well as in the kidney areas where we want to reabsorb water. We will insert more aquaporins into the membrane, and that will help us to bring water across the membrane. If we do not want to actively reabsorb water, again, in the large intestine or in the kidneys, we can remove or downregulate the presence of those aquaporin channels and decrease the water reabsorption. Another type of ion channels, and those have several flavors. You can have leak channels, which freely move ions across. You can have gated channels, which require a ligand to bind to open that channel. Or you can have bidirectional channels, which allow the movement of ions in either direction. Now, factors that affect the rate of ion transport are going to be the transport rate of each channel. Right, the transport rate of a single channel, again, that will vary depending on the tissue location, function of that cell, and so on and so forth. The number of channels in the membrane, as well as the open or closed state of that channel. Okay, specifically, if we talk about, let's say, gated channels, they have to be in an open state, an open configuration to allow the movement of ions. If they're in a closed configuration, they will not allow that.